Hey everybody, we're going to take a look at evidence for evolution today. We're going to look at several different pieces. The first is direct observations, and we're going to see a couple of examples. The first example is going to be color pattern in guppies. This is a field experiment from your book, so you can definitely read all about it. But what we are looking at is how, when changing the selection pressure on male guppies, results in a color pattern change within 15 generations, which is actually less than 2 years. So in this example, you have three pools. Pool 1 has a predator called killfish. It's going to prey on juvenile guppies, and so these juvenile guppies do not express the color genes. So when you look at the adult males, they tend to be much brighter because the predator is going after drab colored or plainly colored fish. In this pool, you've got a different predator. This is the pike cichlid. It's going to be preying mainly on adult guppies. Adult guppies tend to be more colorful, but if you're colorful here, you'll get eaten. So the male guppies tend to be much more drab in color to try and sort of camouflage themselves. Now we have another pool. Here we've got killfish, but no guppies. So what happened was the guppies were taken out of this second pool and transplanted to the third pool with the killfish. So now the killfish are going here after the drab colored fish, and we just transplanted drab colored fish to this pool. And so what we were looking for is there going to be a color change? And the results are down here. And in these graphs, you're looking at both the area of colored spots and the number of colored spots. And you can see that in the source population, the number is fairly low for both. And in the transplanted population, the area and number of colored spots increases. And so what that tells us is that when the fish were transplanted here, the selection pressure changed, and so you started to see adult fish that were more colorful. Now we're looking at HIV drug resistance. So you've got drug resistant strains that were selected for by treatments. And so the result was that resistant strains became 100% dominant in 4 to 5 weeks. So you've got 3 different patients. They all started out, their HIV was very little of it was resistant to 3TC. With continued use of 3TC and replication of the HIV that passed on the resistant genes, we now see that there's quite a bit of resistance. It's up at 100% by week 4 for 2 of the patients, and then the last patient makes it to about 8 weeks. Next, we've got beak size. This was a field study that measured the beak size of birds in a population over several years, and the results show that drought and food competition increased beak size. So in dry years, beaks tended to be larger, whereas in wet years, beak size decreased. Now, more evidence for evolution, another type of evidence I should say, is going to be fossils. So fossils are relics or impressions of organisms from the past. They're really good for showing changes over time from simple to complex. However, many fossils don't have descendants. Now, from an evolution viewpoint, fossils are important because they show how life has changed over time. However, many species fail to, many species fail to survive and become extinct. However, there are a few things we need to talk about. Number one is that fossilization is a rare event. And even when things do become fossilized, only hard parts fossilize well. So soft tissue doesn't fossilize well, and so it's hard to find. Fossils in general are fairly hard to find, so scientists have a very good idea a lot of times of where to look, but they're sometimes not on the surface, so you have to dig for them. And even if they are on the surface, you don't know precisely where to find them, so there's some luck involved there as well. Next, fossils have to be interpreted, so it's not always clear what they are. You don't always have the entire fossil of a full organism, so there's a little bit of interpretation that goes into that as well. And 5 says missing links, and so what that means is we have this fossil record that's not totally complete, and so there's areas where you see jumps from a fairly simple organism to one that's quite a bit more complex, and you know there's steps that are missing in between. Next we have homology, and homologous structures show a common building plan but with divergent functions. And so what we're looking at here is the arm of a human, a cat, a whale, and a bat. Okay, and so you can see by the highlighted bones that the number and location of the bones is pretty similar. They are put together a little bit differently in some cases. But then I should say the number of types of bone are going to be very similar. The actual number of bones varies, but what you'll see is that they're all color coded and they sort of work out to fall in roughly the same place. It's just the function of these limbs that have changed. Problems with homologous structures, sometimes you see vestigial organs, and these are going to be rudimentary structures that a lot of times don't have any use. And so an example would be whale legs or a human tail. So from the evolutionary standpoint, this shows that remodeling of ancestral structures has essentially changed the structures as their functions or adaptations have changed. We also have homology in embryos, though, and this shows that closely related organisms go through very similar stages during their embryonic development. And so here what you're looking at is a chick embryo and a human embryo. And if I was not labeling them for you, I don't know that you would really be able to tell them apart because they do look very, very similar. The chick doesn't look very much like a chick. The human embryo doesn't look very much like a human, but they do look very similar to each other. And so what this shows us is that ontogeny is a replay of phylogeny, or development reflects descent. So essentially when you look at organisms that are fairly closely related, so like all vertebrates, are going to develop in a similar manner. Next we've got convergent evolution, and this is when unrelated organisms show similar adaptations. And this is usually caused when organisms live in similar environments with similar selection pressures. So here the example is you've got a sugar glider and you've got a flying squirrel. These two organisms look very, very similar. However, a sugar glider is from Australia and a flying squirrel is from North America. So they are not very closely related. However, they live under similar pressures, and so they have developed fairly similar structures because it allows them to be successful in their environments. Next, we've got biogeography, and this is the geographical distribution of species. Some things that we look at are species mixtures on islands, and a really good example is marsupials in Australia. So from an evolutionary viewpoint, biogeographical patterns reflect descent from the ancestors that colonized the area. So here we're looking at Drosophila, so that's a fruit fly, and we're looking at how it migrated and evolved as it went from one Hawaiian island onto the others. We also can look at molecular biology. So this is the study of evolution at the DNA or protein levels. And what we see is that related species have similar DNA sequences. Here we have a breakdown of, human at the top, and we're comparing now the amino acid sequences for human hemoglobin polypeptide. So we have, we're looking at human, so there's zero differences. But now we're comparing it to all these other organisms. So what this tells us is that we have more similar hemoglobin to a mouse than we do to a lamprey, which would tend to make sense. So the evolutionary viewpoint for this says that related species share common ancestral DNA. So the closer the relationship, the more similar you would expect the DNA sequences to be. All right, so your summary for chapter 22. You guys should know Darwin's idea of descent with modification through natural selection, which is now our theory of evolution. Notice that theory is capitalized. You should understand that predictions of a theory are tested by experiments and observation, so that scientific theories, are very well tested and well supported within the field. You also need to be familiar with the pre-Darwinian views of evolution, or lack thereof. You need to know Darwin's observations and inferences, and you should also be able to discuss the different pieces of evidence of Darwinian evolution. So that's what we just talked about today. And so for your lecture question, I just want you guys to explain to me in your own words how fossils are used as evidence for evolution. All right, so shoot me a message with your answer, and I'll see you later. Bye, guys.